Okay, then in this video, we are going to talk about glycolysis. That what is glycolysis and in how many steps it occurs. So let's talk about glycolysis process. So what is glycolysis? As you can predict from its name, that glycolysis is consists of two words. One is glycos. These are the Greek words. One word is glycos. Second word is lysis. What means glycos? Glycos means nothing but glucose. And what is lysis? Lysis means breakdown. So the process of breakdown of glucose is called glycolysis. And when does this occur? This occurs in the cytoplasm during the cellular respiration in all the living beings, whether it is plant or animal, whether it is human being or microbes. In every situation, this process of glycolysis occur to break the glucose into the pyruvic acid or pyruvate. So the process of breakdown of glucose, as we know that there are how much molecules are in the glucose? There are six atoms of carbon in the glucose. And how many atoms of carbon are there in the pyruvic acid? 3. So how many pyruvic acid will be produced from one glucose? It will be 2 pyruvic acid. So the breakdown of glucose, that in which step this 6 carbon glucose breaks into 3 carbon pyruvic acid is known as the glycolysis. So we are going to discuss about this. So where does this occur? This occur in the cytoplasm of the cell. As we know that respiration is of two types. What is respiration? Obviously it is not inspiration and expiration, because inspiration and expiration is called breathing. Okay, inhaling of gas and exhaling of gas is not respiration. In the true sense, it is breathing. So what is respiration? Respiration is breakdown of nutrients like glucose, protein or fat, and to obtain energy from this in the multiple processes, and as a byproduct CO2 and H2O also gets released. So this process is called respiration. As we know that it occurs in the two steps. One occurs in the presence of oxygen and one occurs in the absence of oxygen. Which occurs in the absence of oxygen is called anaerobic respiration, and which occurs in the presence of oxygen is called aerobic respiration. This anaerobic respiration, which occur in the absence of oxygen, it may be like lactate fermentation or alcohol fermentation. But in this video, we are going to discuss about glycolysis. Glycolysis is a common step, whether it is lactate fermentation, alcoholic fermentation, or aerobic respiration. Glycolysis must occur before these processes. So let's talk about glycolysis. So what happens? Firstly, glucose enters. As we know that food digestion gets completed in the intestine, in the ileum part. It gets absorbed and it's taken back to the cell. When it comes to the cell, firstly glucose enters into the cytoplasm, and this glucose now breaks in the pyruvic acid in the cytoplasm of the cell, and then it is transferred to the mitochondria, where complete breakdown means aerobic respiration occurs. So glycolysis is just breakdown of glucose into the pyruvic acid. So firstly glucose, which is a 6 carbon molecule, is taken to the cytoplasm, where an enzyme called hexokinase works on this. Hexokinase works on the glucose and it converts the glucose into glucose 6-phosphate. Glucose 6-phosphate. Actually, glycolysis is the 10-step process. In the 10-step, glucose breaks into the pyruvic acid after the reaction of 10 steps. So firstly, on the glucose, hexokinase works, and hexokinase breaks the glucose into the glucose 6-phosphate, and in this process ATP is also used. ATP is used here. Okay, ATP is used here and it forms ADP plus inorganic phosphate, and this inorganic phosphate is added at the 6th position of the glucose and it forms glucose 6-phosphate. This glucose 6-phosphate then isomerizes into the fructose 6-phosphate by the help of enzyme called isomerase. Isomerase enzyme works on this, which converts glucose 6-phosphate into fructose 6-phosphate, and this fructose 6-phosphate is now, so this is the first step, the second step. In the third step, this fructose 6 phosphate is now converted into the fructose 1,6 biphosphate. Fructose 1,6 bisphosphate, you can say. Fructose 1,6 bisphosphate. So where does another phosphate comes from? This phosphate again comes from the decomposition of ATP. ATP breaks and it forms ADP plus inorganic phosphate, and this phosphate again gets added at the first position of fructose to forms fructose 1,6 biphosphate. This fructose 1,6 biphosphate, and which enzyme works here? Here enzyme works phosphofructokinase. Which enzyme? Phosphofructokinase. Now all the molecules are 6 carbon, like this is also 6 carbon molecule, this is also 6 carbon molecule, this is also 6 carbon molecule. Now this fructose 6 phosphate just splits into two parts by the help of enzyme aldolase. Which enzyme works here? Aldolase, which breaks the fructose 1,6 biphosphate into two parts. One is called dihydroxyacetone phosphate, or it is also called, you can say in short, DHAP, DHAP. And this dihydroxyacetone phosphate, this dihydroxyacetone phosphate is in the equilibrium condition with 3-phosphoglyceraldehyde. Which molecule this is? 3-phosphoglycerol. Is it visible? Yes. 3-phosphoglyceraldehyde. 3-phosphoglyceraldehyde. You can say in short 3-PGAL. Okay, and this is DHAP. So these two molecules get formed. So how many atoms of carbon will be in this molecule? That will be 3 carbon. Okay, it will be 3 carbon. Since it is 6 carbon and it has been split into two equal parts, one is called dihydroxyacetone phosphate and this is called 3-phosphoglyceraldehyde and also called 3-PGAL, and these both are 3 carbon molecules. Now what happens? Now I am considering only that this 3-PGAL only formed is 2 molecule gets formed, because this also has been converted into this part. So I am taking 2 molecule of 3-PGAL, means 3-phosphoglyceraldehyde. So what happens? There's this 3-phosphoglyceraldehyde, and how many molecules I am considering? 2 molecules. Why 2 molecule? One molecule is this and one molecule comes from here. So this phosphoglyceraldehyde gets converted into 1,3-bisphosphoglyceric acid. And what happens here, that in this situation the enzyme which causes is dehydrogenase. Is what? Dehydrogenase. And in this situation also NAD plus gets converted into NADH plus H plus. And as we know that what is NADH? It is nicotinamide adenine dinucleotide hydrogenase. So NAD plus gets reduced because hydrogen gets removed from here, and as we know that removal of hydrogen is called what? It is oxidation, and by the oxidation of aldehyde which gets formed? Acid. When aldehyde gets oxidized, then what gets formed? Acid. So here the process of oxidation is occurring, and that's why aldehyde is converting into the glyceric acid, and you can say in short like BPGA. Okay, and from where again a phosphate molecule is coming? From where the phosphate atom is coming? It is coming from H3PO4, means phosphoric acid. From phosphoric acid this phosphorus molecule is coming, it is added to the phosphoglyceraldehyde and is forming 1,3-biphosphoglyceric acid. Now what happens, that it releases one phosphorus means ATP, ADP gets joined with one phosphate and it forms ATP. And how many molecules are there? There are two molecules. There are two molecules. Since there are two molecules, so how many ADP will be converted into ATP? It will be also two molecules. Okay, and it will form what? It will form 3-phosphoglyceric acid. 3-phosphoglyceric acid, or you can say 3-PGA. 3-PGA. Okay, 3-PGA. Now what happens? This 3-PGA, and which enzyme is working here? Here the enzyme which is working is phosphoglycerate kinase. The enzyme which is working here is phosphoglycerate kinase. Okay. Now this 3-phosphoglyceric acid gets converted into 2-phosphoglyceric acid. Okay, means what is happening here? Mutation is occur. That's why the enzyme which is working here is mutase, and it is converting 3-phosphoglyceric acid into 2-phosphoglyceric acid. Nothing is happening, but just it is mutation. Phosphoglyceric acid. And now what will happen, that in this situation again H2O will be removed from here, and if H2O gets removed means enolation is happening here. So the enzyme which is occurring here is enolase, which is causing removal of H2O and converting it into 3-phosphoenol pyruvate. 3-phosphoenol pyruvate. And on this 3-phosphoenol pyruvate dehydrogenase works. Pyruvate dehydrogenase works, and which converts 3-phosphoenol pyruvate, you can say in short like 3-PEP, which about which we have to study about in the photosynthesis process, that in the C4 plants PEP is also present, which is the primary CO2 acceptor in the mesophyll cell, and from where this combines with the CO2 forms OAA, oxaloacetic acid, which gets converted into malic acid, which taken back to the bundle sheath cell where it decarboxylation occurs and it releases CO2. That CO2 enters into the Calvin cycle, and again this pyruvic acid gets back to the mesophyll cell where it's converted into PEP. Okay, these are all the mechanism of the C4 plant in the photosynthesis process. So this gets converted into pyruvate or pyruvic acid. Pyruvate, or you can say pyruvic acid. How many molecules will be formed here? There are two molecule. So this will be also two molecule. 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 So how much molecule of pyruvate has been produced from one molecule of glucose? From one molecule of glucose, there are two molecule of pyruvate is getting formed, and this whole process is known as glycolysis, which is completely an enzyme derivative process. At each step, enzyme is working first hexokinase, then isomerase, then phosphofructokinase, then aldolase. Again here dehydrogenase, here phosphoglycerate kinase, here mutase, here enolase, and here again pyruvate dehydrogenase. And this is a process which occurs in every organism, whether it is plant, whether it is animal, a human being, or microbes. In every individual, this process of glycolysis occurs. Okay, listen once again. Firstly, glucose adds phosphate in the presence of enzyme hexokinase and gets converted into glucose 6-phosphate. In glucose 6-phosphate, isomerization occurs, and as we know that glucose and fructose are isomer of each other, so glucose 6-phosphate is getting converted into the fructose 6-phosphate. And you can note here that this step is not happens if the substrate is fructose. What is substrate? We know that this fatty substrate are those substances which gets enters into the respiration process. Like here, glucose is the fatty substrate. And if this fatty substrate is fructose, then in this situation there is no need of the isomerization, and so this process means second step is not present, does not occur if substrate is fructose. So fructose 6-phosphate, which again adds a phosphate in the presence of enzyme phosphofructokinase and gets converted into the fructose 1,6-bisphosphate. Then it splits by the help of enzyme aldolase and forms dihydroxyacetone phosphate with the equilibrium with the 3-phosphoglyceraldehyde. And now we are considering the 2 molecule of 3-phosphoglyceraldehyde and 2 molecule of 3-phosphoglyceraldehyde. Dehydrogenation occurs in this case, and also this hydrogen which is removed from this molecule 3-phosphoglyceraldehyde is taken by the NAD plus to form NADH plus H plus, that is nicotinamide adenine dihydrogenase. And to add the phosphate we add H3PO4. H3PO4 phosphorus gets added at the first position and gets converted, and since hydrogen is removed from here means oxidation is happening here, and we know that by the oxidation of aldehyde which gets formed, by the oxidation of aldehyde acid gets formed, that's why it is getting converted into the 1,3-bisphosphoglyceric acid, and then it releases inorganic phosphate which is accepted by the ADP to form the ATP in the presence of phosphoglycerate kinase, and it forms 3-phosphoglyceric acid. Then mutation occur, and this 3-phosphoglyceric acid gets converted into the 2-phosphoglyceric acid, from where enolation occur in the presence of enzyme enolase which gets converted it into 3-phosphoenol pyruvate, and this 3-phosphoenol pyruvate in the presence of enzyme phosphodehydrogenase gets converted into the pyruvate or pyruvic acid which is 2 molecule. Now note here that what is the net output of ATP after glycolysis. So you can note here that 2 ATP is being consumed. So ATP consumed is 2. Okay, is it visible? ATP consumed is what? 2. If you talk about how much ATP produced, so direct ATP production is, you can see here this is direct ATP production, and how many ATP? 2 ATP. And again in this process also, when 3-phosphoenol pyruvate gets converted into the pyruvate, then here also ADP gets converts into ATP. So how much molecule here again? 2 molecule. So 2 molecule and 2 molecule, this is 4 molecule. And if you talk about NADH plus H plus, so how much molecule of NAD is getting formed? You can see here 1 molecule, because 2 molecule, because you can say this is 2 molecule means all the processes are happening twice. So this is also 2 molecule. And we know that 1 NADH produces 3 ATP after getting entered into the ETS means electron transport system, means here 6 ATP will be produced, means net production is, total production is 6 plus 4 10, and consumption is 2, means total production is 10 ATP, consumption is 4 ATP, sorry 2 ATP, consumption is 2 ATP. So what will be net ATP production? It will be 8 molecule of ATP is getting produced in this situation, and this is a net output of glycolysis, and it happens in everywhere, whether it is plant, animal or microbes. So 8 ATP gets produced after glycolysis, in which 3 gets formed, 6 gets formed as NADH, nicotinamide adenine dihydrogenase. Okay, so you can take a screenshot or you can write it by pausing the video, and you have to write each and every enzyme, and if you practice it multiple times, more than 2 times, you will be able to learn it also. So okay, just finish this video. We'll talk in next video about the different type of fermentation and also aerobification like Krebs cycle. Till then, keep studying. If you want, you can take a screenshot, and we'll talk in next video. Till then, keep studying. Okay, thank you.